Hello and welcome to the IE2 seminar. This week, we are very pleased to have Lauren Ponisio coming to speak to us from the University of Oregon. I think she's well enough known to everybody. I'm not going to list through her accomplishments and just say that we're really happy to have her. So you want to take it away, Lauren?
Sure. Thanks, Peter. Okay.
Thank you.
Here. Hi, everyone. I'm Lauren. Ooh. Maybe mute yourself, Peter. Great. So nice to meet everyone. I'm at home, but soon I will be in my office in the third floor of Onyx. And today I'm going to talk about host traits, microbes, and disease in intensively managed agriculture. And, just another hello, we arrived last July in secret, kind of secret, mostly pandemic, and really looking forward to meet everyone. This is the current members of the lab right now. Many of you have probably met Nicole or Rebecca. They're new first years. Jess is a master's student starting in the winter. Jocelyn is our lab manager. She's the person who's always in Pacific 221 if you ever see her, crouched over bees. And Emmanuel is a visiting scholar from Brazil. So, super excited to get to meet everyone and see everyone. We have a move-in date for our lab. It's November 1st. I've been thinking about what fun thing we can do to get you guys over to the third floor of Onyx, so far from the third floor of Pacific and have some kind of fun something. Well, we also had other lab managers who are lab members who were here last year who have since moved on, and they are the ones I wanted to highlight, particularly for this talk. Dr. Helmantal Cohen, she accepted a job in Florida. Jenny Durant, now in Washington, D.C. Gordon Smith, now finishing a NSF postdoc at American Museum, and Jocelyn's still around. So Jocelyn's the one lab member involved in a lot of this work who's still about. So many of you guys are probably aware, particularly given people's research interests, that microbes and wildlife, there's this growing recognition for the importance of microbes for health, particularly concerning disease susceptibility, reproductive fitness, and a variety of different disorders. And people tend to contrast the pathobiome, so the kind of the biome that makes the microbes that make you sick from the beneficial kind of happy microbiome. But people have moved past this idea of one pathogen is equal to one disease and really thinking of this whole pathobiome. So it's not, often sometimes you do get one pathogen and then you do get a certain disease state, but a lot of times it's the complementary and synergistic interactions between lots of pathogens and potentially your microbiome that lead to all the problems that we have. So that leads to the question, well, then why do microbes vary between hosts? Can we understand in some way both the pathobiome and the beneficial microbiome, and how different types of hosts have different compositions of these microbes? We can look at ecology and community assembly theory generally and think about, what do we have that's really well established in ecology? And this idea of habitat filters, being deterministic of community composition is pretty well established. So you have some regional species pool out there, like all bees or all the other species that I never think about, plants are out there. There's some biotic or abiotic filter. This can be habitat type or different kinds of species interactions, and that's going to lead to some kind of local differences in local community composition. And so let's think, maybe in the world of microbes, it's less about habitats and more about the hosts and the type of habitat they provide, which might be influenced by their different traits. So you have some environmental microbiome pool, then you have some host traits, and depending on the host traits, different microbes might colonize these different hosts. And so for the first part of the talk, I'm going to ask, how do host traits affect the beneficial and microbiome and pathobiome, basically asking, is this conceptual framework accurate? And I'm going to focus on bees. So bees are an amazing group of species. They come in all sorts of shapes and sizes. There are about 1,500 to 1,700 bees in California, where we were working at the time when we did this project. Actually, California is a global hotspot for bee diversity. So bees are a group that do not follow the latitudinal gradient in diversity. They like Mediterranean environments like California. And actually, Oregon has about 500 species of bees, so not too shabby. And there's this amazing project going on called the Oregon Bee Project, where they've chronicled just all the bees of Oregon, have incredible pictures, incredible ID guides. If you guys want to learn more about bees, check out their website. It's such an awesome resource to be in a state that takes bees so seriously. And of course, we also have the European honeybee. So the European honeybee is not native to the Americas. It was originally introduced by the Pilgrims and has really stuck around as part of our industrial agricultural system. So it's not one of our native bees, but it's also a bee, so we tend to include it in our communities. And bees are incredibly important because of crop dependency on pollinators. So different crops have different levels of dependency. Something like sunflower is 100% dependent. So every single sunflower seed is the result of an animal moving pollen from one flower to another. Something on the other end is like grapes. They don't really need much, they don't need any pollination, which I think is a real tragedy. I would love to work with Krista in wineries, but sadly, biology is against me. But really importantly, my favorite things in the world are berries, like blueberries, and those have a lot of pollination dependence. And in general, US animal pollination is worth about $14 billion each year. It's about a third of our production. So particularly for all the fruits and nuts and things like that. Cereals are really the only major crop group that are not animal pollinated. So that's like corn and wheat. And those are all just giant grasses. So they're wind pollinated. And because it's mostly the fruits and nuts and delicious things, the majority of our nutrients come from animal pollinated crops. And bees are in decline. So particularly from pesticides, inadequate nutrition, and disease, which we'll be talking a lot about today. This is a really cool picture of the US. And you see, when you see those big bright red squares, that is where there's really high demand for pollination and really low availability of bees. So particularly the Central Valley, where we worked, is a place where there's lots of animal pollinated crops. And because of industrial agriculture, not very many bees. So starting with the first part of the talk, we're going to talk about how these host traits affect the beneficial microbiome and pathobiome. And then for the second part of the talk, given the importance of agriculture and disease for bees, we're going to talk about specifically the pathobiome and how does agriculture affect parasitism in wild bees. So, but first, to really kind of get what's going on with all these things, I'm going to tell you a ton about bees. And this is adorable little sunflower bee scratching her head and pondering life and the world. So first of all, how are microbes transmitted? And in the bee world, microbes are usually transmitted horizontally. So instead of vertically, like mother to daughter, most microbes are transmitted between bees, particularly via flowers. So you can imagine going out, a bee visits a certain flower, they have a certain microbe associated with them, and then they either pick up new microbes or deposit microbes on the flower via defecation. So, basically flowers are the bathroom doorknobs of the bee world. Bees will often defecate in flight or while they're foraging. A lot of the pathogens they have and parasites will actually make them have like bee diarrhea. So they'll be more likely to defecate on flowers. Then they'll actually track that, around the flower as they forage and look for nectar. This is a photo out of the Rankin-Wilson lab where those little tiny red footprints are actually bee feces being moved around. They found a way to photograph this. And so yes, bees are bathroom doorknobs. That is where all the parasites they pick up often are from flowers. So hopefully did not totally ruin flowers for you, but for bees, they're a risky place. And we can also think, if all these parasites are coming from flowers, depending on what flowers you're visiting, this is kind of like a corridor, sort of like facilitating those microbes getting into the bees, because they're really picking them up on the flowers. So if you're visiting lots of different flowers, is this kind of like the corridor width, like a really large corridor, lots of microbes getting in, versus a more specialist bee that's visiting only a few flowers. Maybe that's like having a really narrow corridor. There are other ways to pick up microbes as well. So living together is one of them. Most bees are solitary, so they have, a female bee will go out, do her foraging. She'll build a nest primarily underground through repeated foraging visits for pollen and nectar, lay a little egg, little larva, pupa, that cycle continues. So each female bee makes her own nest. So there's not a lot of vertical transmission in this case. However, there are some social bees like bumblebees and the European honeybee that live together in colonies. And there are workers that go out, collect food, bring it together, bring it to one place, and then other bees will consume that food. So there's a lot more kind of bee to bee touching as well as transmission through the food resources. Bees also nest in places. And so just like with humans or other organisms, nesting material or like structures are a big way that bees pick up microbes. And bees nest in some of the craziest, weirdest different ways. Some of them will collect fuzzy stuff from leaves to line their nest with. These are wool carder bees. It's like a little female with a little like wool ball. Others will actually cut into leaf material. So you imagine there's a lot of fun microbes in leaves and line their nest with leaves. Similar species of bees will actually use modified leaves, which are petals instead. Others will nest in pithy stems. So these are actually really common here in the Pacific Northwest because we have so much blackberry and raspberry and things that create pithy stems. And so they'll make nests down those tubes. So other fun microbes associated with those pithy stems. Most bees nest in the ground. And so they're going to be contacting soil on their mandibles and just really getting down and dirty in there. So you can see this little bee popping out of some soil. And there's just all sorts of varieties. So bumblebees will also, nest just in these hollows of trees or like right in the ground. And some will even create these little structures with resin. It's kind of incredible the amount of diversity of nesting out there. But all of this is having them touch, and microbes in different ways. And lastly, we have traits that might affect microbe colonization and persistence. So particularly body size would potentially affect colonization because bees with larger bodies actually disperse longer. So body size is really proportional to wing muscle size. So something like a bumblebee, a really big bee, can disperse a lot farther than something tiny like this little Ceratina, who's one of those pithy stem nesters. Also, there's more area of the gut where all these microbes are hanging out. So there has been some relationship shown between how large of a bee you are and how many parasites you have, just because you have kind of more like it's like more habitat area to go around. And lastly, bees also collect pollen in a bunch of different ways. So some have these really beautiful hairy legs. Others, which is something you might be more familiar with, like bumblebees, will wet collect their pollen onto flattened plates. Some have hairy bellies that will actually kind of like wiggle around on flowers. And there is a whole group of bees that have no external pollen carrying structures. They ingest all their pollen and then regurgitate it. So all of these ways, again, all of these different traits, again, will potentially affect how microbes are able to persist and colonize these different bees. Okay, so that was all the things about bees. You didn't know you were getting into with this talk, but now hopefully you guys will abandon whatever study systems you have and join the bee world. So that was my hope. But getting back to thinking about ecology and host traits, we have all these traits. We are generally aware that they might affect microbe acquisition, but how do we quantify how much of a filter host traits might impose? And we can borrow some methods from community ecology, where if you think of, there are these PCOA, so principal coordinate axes, in n-dimensional space. And you can think of each dimension as one of these traits I just went through. And then we have these different bees kind of plotted on a flat plane in that n-dimensional space. And if we plot kind of the middle, like this is the hypothetical middle trait value, the more common trait value in the community, if we look at the distance from that centroid or middle value, we can think of that as host trait distinctness. So people have used this in different contexts to study, for example, differences in community composition or trait diversity. But here we can think of this as, how basically weird or unique a host is in its traits. And so when we think of that conceptual framework, if we're looking at the thickness of this filter, you can imagine putting host trait distinctness on this axis. So species with super distinct traits are potentially, it's more difficult for microbes to colonize them. Whereas something with really common traits is really abundant in the community, then this is maybe not so much of a filter and lots of different microbes can colonize it. We can also use the exact same method to look at the community composition of the microbes. So instead of imagining a matrix with all the different species of bees and their traits, imagine those traits are the microbe species. And so some species might have really unique microbiomes, whereas others might have really common ones. Does that make sense? Okay. So first part of the talk, thinking about how do host traits affect the beneficial microbiome and pathobiome. And I'm going to also begin with thinking about the study system we worked in. So we worked in sunflower, which I mentioned earlier is 100% pollinator dependent. And there is an extremely high density of bees in sunflower. It's a huge nectar and pollen resources with a whole variety of life histories. And now you can just imagine all of that covered in little microbes. We worked in a system where we also kind of wanted to make sure we were getting enough variety of life histories to be able to look at this trait distinctness. So we worked in places with hedgerows. So this is the plus HR, and also just weedy field margins, like stuff that was just out there. And we did a full cross design where we had some sites with sunflower, some sites without sunflower, some sites with hedgerows, some sites without hedgerows, and track them all through the season. So the idea again is to get a variety of different bees from different life histories, early in the season, late, and during the bloom. This is what the Central Valley of California looks like. So we're over here in basically actually right by Davis. So Davis, it's like kind of over here. This is woodland. And you can, our sites are kind of spread out over this system. And sunflower is actually rotated through years. So sunflower is never placed in the same place, same crop field twice, because they don't want the F1s to interbreed with the parents. And so they kind of constantly move this around. This will become more important in the next section. There is some natural habitat, but it's not super, doesn't seem to provide much for bees in this system. And you can see there are kind of hedgerows scattered around the landscape as well. And what's really great about sunflower as a system is, first of all, that we have sunflower specialist bees. So sunflower is a native North American crop. And so we have our own sunflower bees that actually nest in these little holes. This is a picture that I took just looking down at the base of a sunflower crop field. And you can see all the beautiful sunflower pollen. So these are where the bees nest. And you can see just in one sunflower, there's this really kind of large bodied sunflower specialist that's solitary. This is a tiny little small bodied bee that's social with an extremely generalist, really wide diet breath. And of course, the European honeybee, which has a very, very wide diet breath, medium bodied and social. So all of these different bees will visit sunflower. And so our hypotheses for the system to address how host traits affect the beneficial microbiome and pathobiome were that host trait distinctness will be positively related to both microbiome and pathobiome distinctness, and that individual diet breath will be negatively related to microbiome and pathobiome distinctness. This is again that idea of the corridor. So the larger the diet breath, we expected that would lead to lower distinctness of the microbiome and pathobiome. And we use sterile collection methods. This is my colleague, Hollis Woodard, out there with her gloves. We carry around bleach and ethanol, and we just kind of constantly re-sterilize, collect all the bees into individual sterile tubes, put them on dry ice, bring it back to the lab, gut them, and analyze them for a variety of things. So, and particularly, we collected about 1,600 species of bees and managed honeybees across 34 different species. We use 16S metabartic coding to quantify their microbiomes. We also screened them for 7 common bee parasites, and these are a whole variety of different little creatures. And we removed pollen from their bodies and used metabarcoding to figure out which species of plant that bee visited before we captured and murdered it. Basically their individual dye breath. And I say we, but just to be really clear, this was Gordon, Homental, and Jocelyn. I did help out a couple times, but they put me back in my office after a while. So these are the people who did all this hard work. It took about 3 years of lab work. And for those who are interested, these are the common parasites of bees. We have Crytheria, which is a trypanosomatid. It has all sorts of negative effects on bees. We have the nosemas. These have gotten a lot of attention because they seem to be very associated with bee decline. They cause bee dysentery. It's a little microsporidian. We have a fun little fungus called Ascosphaera that causes a variety of awful things to happen to the broods, and Apocystis, which is whatever one of these thingies is. It can cause a lot of problems with colony function. Okay, so our results. How do host traits affect the beneficial microbiome? We have host trait distinctness on the x-axis, and we have pathobiome distinctness and microbiome distinctness on the y. And as we expected, pathobiome distinctness seems to be positively related to host trait distinctness. We did not see that in the microbiome. So one piece of this meets our hypothesis, and the other maybe not so much. Then we want to know about that kind of corridor question. So what about individual diet breath? Is individual diet breath, we expected, negatively related to pathobiome distinctness? Not so much, pretty flat line, about as flat as you can get on that one. But it was positively related with microbiome distinctness. So we don't tend to see in individuals diet breath is strongly related to pathobiome distinctness, but it is for the microbiome. It's kind of interesting that, the pathobiome and the microbiome seem to be responding to really different things. So going back to our hypotheses, we see that yes, indeed, host trait distinctness is positively related to just the pathobiome distinctness, but not so much the microbiome. And the reverse of what we thought we're expecting, which is that individual diet breath was actually positively related to microbiome distinctness and was no real strong relationship with the pathobiome. It's a bit of a head scratcher on that one, but we pondered it and we came up with a potential explanation for it. Basically, instead of this idea of diet breath as a corridor, we know that bees have patterns in their interactions. And it's not just, generalists visit these species and specialists visit these. They tend to see what's called asymmetric specialization. So this is basically that, for example, a really generalist bee, like this bumblebee, tends to visit a whole variety of flowers, including specialists and generalists, and vice versa from the plant's perspective. So if you are a species with a really wide diet breath, you're picking up microbes from all of these different flowers. And potentially, maybe the specialist flowers have a different suite of microbes. And so in that way, you actually end up with a really distinct microbiome versus what we were originally expecting, which was just this simple kind of corridor idea. So that's our proposed mechanism for why that might be happening. So it does seem in general that this idea that host traits are a filter is plausible, and particularly for the pathobiome, and that at least for the microbiome, that diet breath is really, really part of that. And so implications for habitat loss and continued global change is that we are losing host diversity. And so this might be basically the same kind of a corollary in the microbe world to losing host traits. And so, habitat loss leads to host trait diversity loss. Host trait diversity loss seems like it will also lead to microbe diversity loss. So another reason to not do that. Okay, so that was the first part of the talk, talking about the beneficial microbiome and the pathobiome. And then for the second part, let's zoom into that pathobiome and really try to understand how does agriculture affect this parasite prevalence. And so zooming in to, the sad spiky microbes that cause all the problems. And going back to this crop dependency on pollinators, we can break down each of these little pieces into percentages. So there's some species that really have this essential, pollination is essential for them all the way down to little. And the majority of these species that fall in this bracket are what are called mass flowering or mass blooming crops. And basically it means that, everything blooms at once. That's actually, as you can imagine, if you were a crop, that's kind of the way to go, because then there's lots of pollen out there being moved around. And so we really wanted to understand, how does this big pulse of blooms, which provide nectar and pollen for bees, but, might have other effects like increasing parasitism, affect their populations. So, think of this beautiful mass blooming crop like sunflower. And again, now it's just tons of bathroom doorknobs, in incredibly high abundance. And remind you guys again that, bees are picking up these parasites from the flowers, and that lots of these parasites give them bee diarrhea, which makes them defecate more on the flowers. So we want to ask, how does agriculture affect parasite prevalence? Our hypotheses were that through augmenting bee abundance, mass flowering crops would amplify parasitism. So this classic, host abundance, host density leads to more parasitism. But that on-farm diversification, habitat diversification, particularly something like a hedgerow, would dilute parasitism by breaking up the chain of transmission. So another classic hypothesis in disease ecology, that's, if you have a diversity of hosts or a diversity of, in this case, flowers, which are kind of, intermediaries between the hosts, that this will potentially break up transmission. And our study system, again, is we have those mass blooming crops with or without hedgerows, and we track them through the whole season. Now, looking at this map, there's an important thing to point out with the rotational nature of the crops, is that because the sunflower bees actually nest at the base of the crops, they appear to track the sunflowers around the landscape. So if you go to a place where there was sunflower in the previous year, right before sunflower starts to bloom, you suddenly see all of these bees just emerging from the ground and kind of somehow figuring out where the sunflower has moved and showing up there. So we were interested in also, this rotational nature and how that would affect the distribution of bees in the landscape. So we surveyed these are the bees we actually caught and captured. We surveyed about 3,400 bees or so, 15 genera, 34 species. These are the same bees we screened for the other project, but we didn't focus on honeybees in this project because they are managed and seemed like they'd be having really different responses than the wild bees. Only 9% of them had no parasites. Most of them had at least one of the parasites we screened for. And a couple, 2 to 3%, or 35% had 2 to 3 parasites. So a lot of parasitism in this system. So really pretty high parasite prevalence. And so asking, do mass flowering crops enhance bee abundance? We looked at this 2 different ways. One was looking within a year, looking at that pulse of flower availability when the sunflower came into bloom. We expected that would lead to higher bee abundance. And also this idea of thinking about the area of sunflower from the previous year around the focal site. So the idea is that, the more sunflower there was last year, if they're tracking sunflower, they're going to merge out of the ground and show up at the focal site. And if there was lots of sunflower around, you'd have lots of bees. And this is, again, that because they focus on sunflower as their host plant. And we do indeed see this kind of obvious peak throughout the season and almost exactly when sunflower is in peak bloom. There is also more bees in sunflower than areas without sunflower, like the hedgerows in the weedy field margins. So they seem to really be focusing on sunflower. They seem to be tracking the abundance of bees, tracks the abundance of flowers in bloom. And then we also see that depending on how much area of sunflower you had in the previous year, you tend to have more bees around you in the next year. So overall, sunflower seems to be a pretty important resource for these bees, increasing their abundance both between years and within a year. So then the big question is, does bee abundance then amplify parasitism? So we have wild bee abundance on the x-axis and parasitism rate on the y. And we do see a positive relationship, but it's, pretty pretty weak. Kind of, one of those significant but maybe not biologically relevant moments. But what if we add in floral diversity into the mix, that second hypothesis, or floral abundance? And so thinking about, maybe having other flowers around has some kind of effect on transmission. So we have non-crop flower abundance for the different sites. The hashes are the hedgerows, these purposefully planted plants, and the ones without any hashes are the weedy field margins. You can see that though in general, the hedgerows have more flowers, actually the weedy field margins sometimes have tons. These are things like invasive brassicas and things that bees love and produce tons of flowers. So it seemed like in this moment, it didn't really matter if you were a hedgerow or not. It just mattered, that there are all these flowers out there. So when we consider the abundance of flowers into this equation, we get a really cool effect. So in the sites with the highest abundance of flowers, we don't see parasite amplification with host abundance. So it's actually kind of a negative effect. So we see dilution. It's the ones with the lowest abundance of flowers where we see this positive relationship, though not incredibly steep. So suggesting both a role for amplification via host abundance and dilution via floral abundance in the system. So we did see that through augmenting bee abundance, mass flowering crops seem to amplify parasitism, and that on-farm habitat diversification, which also weedy margins happen to seem to have the same effect in this case, not just hedgerows, dilute parasitism by breaking up the chain of transmission. So fantastic. It was a big moment for us. This paper was actually just came out on Tuesday in Proceedings B, so be sure to check it out. So implications for global change. Basically that more mass blooming crops will lead to more parasitism in bees. I can't imagine that sunflower would be any different than something like almond or blueberry or anything like that. Anything that's concentrating these bee hosts in one place, they're picking up parasites via shared flowers, and then can someone mute themselves? Thanks. So it's just another great reason that we might want to do things like installing habitat in agricultural areas. And we have a variety of papers showing that habitat restoration augments bee abundance and richness and helps the colonization of specialists, promotes beta diversity, all of these things. And now it also seems like it supports parasite dilution as well. So another great reason to do habitat restoration. And so with that, we've talked about how host traits affect the beneficial microbiome and pathobiome, and how agriculture affects parasite prevalence. We found that host trait distinctness is positively related to pathobiome distinctness, and that an individual's diet breadth is positively related to microbiome distinctness. In general, mass blooming crops seem to aggregate hosts, amplifying, not employing parasitism, and that non-crop flowers help to dilute that amplification. And with that, I kind of wanted to just like whirlwind tour through other cool projects in the lab. I think we have a little bit of time. So we have a cool project going on in California, looking at how pollinator communities have changed through time. This is in collaboration with Ruben Alarcon down at CSU Channel Islands. Can someone mute themselves that's doing the dishes? I promise it's not me. And so they did these two transects in the northern Northern California and Southern California, and they went from an island all the way to a high elevation peak. And now it's been 50 years and we're going to resurvey them. It took us 5 years to digitize his Fortran cards, which was brutal. But now we have 100,000 specimens from those original surveys to compare our resurveys to. We also have an NSF-funded grant looking at how pollinator communities can be both resilient and highly functional. So really thinking about these different roles that bees occupy in networks and what are the outcomes for both their function, so this is their providing pollination, and their population fitness. Remember all those super inconvenient fires in 2020? Well, one really convenient thing is there's 5 replicated fires in Oregon for us to study in terms of colonization of bees post-fire and how the different sizes and also sizes of high severity and affects their colonization. This is a collaboration with Jim Rivers at OSU and Shalini Jha in Texas and Laura Burkle in Montana. And I'm super jazzed about a project where we're trying to enhance clear cuts. So this is a picture from out on Whitewater Ranch up the McKenzie, and they make these giant burn piles and they light them on fire. And we're thinking of adding plants to those burn piles for restoring bee populations. So imagine instead of these kind of ugly clear cuts full of, blackberries, gorgeous wildflower meadows full of bees. And this is, again, in collaboration with Jim Rivers, as well as NCASI, who's the research branch of the timber industry and the Department of Fish and Wildlife. And lastly, we recently heard, I think it's still kind of secret, but we just got another USDA grant to study bombus and patience invasions in the Pacific Northwest. So there's this very beautiful, but very mean little bumblebee that's moving down from Canada, basically from greenhouse pollination. And it's actually an eastern bumblebee. And so we're going to understand how that invasion is changing the parasite communities and networks of wild bees in the Pacific Northwest. So with that, I'd like to thank all of you. This is, again, our lovely lab. I also wanted to have a shout out to Abby, who did a rotation project last year and dissected a ton of little syrphids that were part of this project. They weren't in this data, but one day we hope to publish on that. And Gilia, who worked on this project thinking about how we could model disease transmission in something like a mass blooming crop system and understand how host traits might affect that transmission. And with that, I'd like to thank everyone. I'm really excited to get to meet people in the department and look out for an email about some kind of shindig following our lab move-in after November 1st. Awesome. I'd love to take any questions. Hey, Becca.
Oh. Okay, I'll go. I was wondering, really cool stuff. So I can't actually see you anymore. So you were looking at in those original PCOAs, you were looking at differences between species. I was wondering if you had any idea of the variation in microbiome composition within a specific species, and if that differed based on species. Are some species more variable than others?
Yeah, and actually that is something that Gordon Smith, the other postdoc on the project, has been working on, really thinking about the structure of the networks and how that contributes to individual variation. So one really neat thing is we found when the networks are really what we call hubby, so like there's sunflower in the middle and then like all these maybe peripheral plant species, that leads to less microbiome variation than species that have a more, less of a dominant kind of plant species in the middle. And so it's really neat. There also seems to be some cool differences between males and female bees, which tend to have really different foraging patterns. So yeah, you should come hang out and work on questions like that. We also have a lot of fun data that I feel like is really underutilized. So if people have cool ideas, really, really interested to hear them. Peter is waving his hands.
Wait, can she hear me?
Yes.
Okay. I had a question about the like the diet breadth, thinking about that, where if generalist bees are visiting the same flowers as specialist bees, could the generalist bees be bringing microbes from other flowers to that one? And then that could be how some of the specialist bees could get additional microbes.
Yeah, I think the key, like you're saying, is basically that asymmetric specialization where the generalist bees are visiting all the different flowers, and then the specialists are maybe just picking up, like you're saying, the common pathogens that the generalist bees have dropped off. So yeah, it's a neat thought. I've been trying to also ponder how to actually look at it experimentally. We can take the microbes off the plants. It's a little tricky and painful. We did not collect the sunflower heads in this system, something I will always regret, but was because they're a crop and the growers did not want us to collect them. Strange. But maybe in future studies, it'd be great to work on something that we could, destructively sample and really try to understand those networks of deposition. Bill?
Hey, Lauren, thanks. Great talk. It's really fun. It's always fun just to hear cool research that's going on. I've got a question about the original ordinations in trait space and microbiome space and the correlations across them. Have you thought about the fact that the organisms are evolutionarily related to one another? So there's a phylogenetic structure. So oftentimes in comparative biology, independent contrasts are used to control for the degree of evolutionary relatedness. And maybe you said that and I missed it, but I'm not sure if that's something.
No, so we pondered this, and it's a bit of a pickle in the bee world because we don't have a good phylogeny.
Oh, yeah.
And what we did do was we looked at each of the groups we had represented, and we looked at their traits and tried to think, do they look like they're phylogenetically structured? And it's kind of crazy, but like in bee taxonomists are splitters at the generic level and lumpers at the family level. So within a genus, there can be tremendous diversity of life histories. And so, for example, in one genus like Halictus, we saw both, social bees with wide diet breadths and solitary bees with more narrow. So the traits did seem to be distributed across what we had of the phylogeny, but it's definitely always a big limitation in the bee world. We have in our NSF project, we have a lot more diversity, and we are working with a lab at the USDA Bee Lab to make a community phylogeny. So we at least have something, but it is a pickle. Thanks for bringing it up.
Yeah, thanks.
Matt.
Hello. That's a great talk. So I was thinking about when you're sampling parasite diversity and also gut microbiome data in the bees. So have you actually looked at correlations between certain taxa and the presence of certain parasites, or diversity when parasite load is, high, things like that?
Yes.
Am I making sense?
We did do some Mantel tests looking at the correlation of the pathobiome community and the bacterial microbiome community, and they were correlated. Plots don't look very good, but they're correlated. We do not really know enough about bee microbiomes and this is maybe something we're going to delve more into in the future, but what specific groups might be related to resistance to the parasites. We do know kind of what the core species are in the bee microbiome, at least from social species, but very, very little has been done on the solitary species. So it's kind of just like a giant open blank slate. So if you want me to send you that data.
Yeah, maybe we can talk. Thanks.
Yeah.
I have a question.
Hey, Peter.
Can you hear me? Okay. I'm wondering, do you know how much during your study the commercial European honeybee hives changed? How much that was constant versus changing?
Right.
The nearby to your fields?
Yes, and conveniently, there's a standard honeybee stocking rate for all of these fields, and all of our growers stuck to that stocking rate. So there tended to be really similar abundances of bees at the different sites. And they plop them there before everything starts to bloom. So honeybees were out our entire sampling season, and then they stick around the whole season. So it's a really tricky thing in the agricultural systems with the honeybees, because they're hyper abundant since they are stocked, but we also can't really, we can collect some, but we can't collect them proportional to their abundances, because A, that is all we would do. Like, they're just so abundant. And B, the beekeepers would be really angry at us and the growers for destroying the hives they're paying for. So we kind of rely just on that. Because there is a standard stocking rate, they tend to be in pretty similar abundances.
Is it at least like the same hives there the whole season, or?
Yeah. So they contract with a single beekeeper with this system, and the beekeepers come, drop off a bunch of hives on a pallet, and then they'll kind of check on them a little bit through the season, but mostly just leave them there until they pick them up after bloom. Yeah, honeybees were nasty too. 85% of the honeybees we screened, we sampled 5 honeybees per sampling, site per sampling survey, and 85% of them had at least one parasite. So those things are chocked full of nastiness. Stillian.
Hey, Lauren, great to hear about your awesome work. I hadn't actually realized it was so heavily focused on the microbiome, which is great. I have a question regarding your ability and your confidence in classifying microbes as part of the pathobiome or not. Like, how obviously there is overlap, but how much is that overlap in your case, and how what is some of the operational approaches you take? Because, for example, for humans, we know that some bacteria can be pathogenic depending on which part of the body they colonize, but in some other instances they're present but not pathogenic. So what is the state of the situation in your case?
Yeah, so with the parasites we screen for particularly, we know that those cause bee diseases, and we don't know, they can just be, hanging out in a host and not making them sick, or, hanging out for a while and then suddenly, melting them or something. Other people have done the painful task of feeding different species of bees the feces of other sick bees, and found that they can get most bees sick eventually. So they are some kind of disease causing agent. Whether or not the individual bees with a parasite positive were sick in that moment is unknown. And these are all, none of those are bacterial, the ones, the 7 we screened for. So they don't show up in the microbiome. However, we are making kind of a big assumption, well, calling it beneficial, or, I just kind of, it's hard to have the right words for it, in that there are definitely species in there we know can be pathogenic in other species. Like, we ended up with a lot of Wolbachia, but we don't really know what they do in bees. And so that's maybe more like a vocabulary. So there's basically there's the 16S microbiome stuff in their guts, most of which we think is good. And then there are these 7 really bad parasites we know we can make bees sick with. So there's no overlap, but there's probably some pathogenic things in the 16S.
Okay.
Yeah. And the micro, I love making networks of things. And my postdoc Hamantal Cohen was like, we were already going to look at bee diseases and microbiomes for this grant. And then she kind of enticed me with what if we did even more bees, and then we could make cool networks of things. And I was like, cool networks, you say? Let's screen all the bees. So.
Thanks.
Peter is waving his hands.
All right. Sort of jumping on, sort of related to the parasites, do you notice any variation in the bees' microbiomes depending on what parasites they were infected with?
So we haven't really gone further than looking at the correlation between the pathobiome community composition and the 16S microbiome community composition. I do think there is a lot more to think about in terms of maybe individual species of parasites and individual bacteria species. But as you can imagine, there's kind of a really large array of species to choose from. And since we just know so little about the bees, it feels like a little bit of just kind of maybe it's a question that would be better asked experimentally. There have been some cool studies done where they wipe out the microbiomes of bees and then they feed them different pathogens and get them sick in different ways. And so I'm wondering if that might be a better approach for answering some of those questions, but maybe not. And in which case you might know what you're doing more than me and you can have some data. Let me know. Swing by my office on the 3rd floor of Onyx after November 1st. Or email me. Awesome.
Well, let's thank Lauren again. Lauren said she'd hang around and do the usual tea time after this. So if we'll, take a 5-minute break, if anyone wants to come back to this Zoom room and chat some more, then you're welcome to. Thank you.
Bye, everyone. Hey, Bill. Bye, Bill.
Can't tell if it's a high wave or a bi wave, right?
Krista messaged me, I'm so sorry that wine doesn't need bees. I am really sorry that wine grapes don't need bees. It's I feel a constant disappointment.
But bee researchers need wine, isn't that right?
No, we do.
And I can't stick around today, but thank you.
Hi, Peter. It's nice to see you.
Yeah, I'm going to make you host as I leave and, see what happens. Did you notice that we disappeared for a minute in the middle?
I did.
Yeah, I had my computer plugged in almost. The plug was like this far out of the computer, and so all of a sudden, bing.
I realized because I wasn't host, I couldn't mute the person that I could obviously see wasn't muted. Maybe I needed to. I
muted it. Yeah. I forgot to make you co-host.
I thought they were only like washing dishes or something. I was like, this could go badly. It's always my terror of like not muting myself in seminars. I don't know.
Yeah. Okay. Thank you. And I'll see you later.
Bye, Peter.
Hey, Lauren, I had more questions if you don't.
Oh, yeah. Well, I'm glad I could like dangle this data in front of you.